We will start this discussion on precision medicine and clinical care. If you say it is added a new chapter in our Harrison 21 edition, but this chapter we were knowing for a long time. Even I can say when I was appearing in exam somewhere in 2014-15, that time also this used to be one of the question to prepare. But things have changed a lot. What I used to write in 2015 seems outdated when I talk about precision medicine. Our understanding of disease is evolving day by day. So it is better to know. No, today we are not practicing precision medicine, but we are practicing few component of precision medicine for sure. There are certain genetic tests and we know, okay, this reaction with particular drug will be more common. That is nothing but the result of precision medicine. So we will try to understand the concept. Like if I tell you abacavir, if I tell you allopurinol, there are certain genetic mutation, right? If we test it and if patient has that, then it is likely that they may develop that hypersensitivity syndrome. So for that patient, that drug is not preferable. That's a precise information what you get with the genetic studies, right? So there are so many layers and component of all this thing, and the information is so much. I'm not sure the human brain can understand and what will happen next, right? But we will try to see what is the concept of this. And meanwhile, we are also preparing a short note of this when it comes to your exam. So there will be so many aspects covered in this next 20 to 30, maximum 40 minutes, that what we are going to discuss about this. So theory exam, it's quite an important chapter. Few MCQ may come from NEET SS. Practical exam, I told that few drugs and susceptibility. Clinical practice, this is the future, probably maybe after 20-30 years. And I hope we all will be practicing at that time. So this is going to come to us. As we can say that 20 years back when people were studying, biologics were new thing. They had no idea. But now those same people are practicing and they have adapted themselves to biologics. Same way we have to adapt ourselves to this precision medicine, right? So it's ever evolving things. MD medicine never gets over in 3 years, never, right? So with this, we will start our discussion. And this is the first slide I want to introduce. If you have communicated to me sometime in life, maybe this guy have answered you, this guy have answered you, this guy, rarely this guy has answered you. Okay. It's all about the digital avatars, right? So we all have, we can't deny the fact now that I do not exist online. We all exist there. And I can say 50% of our life is actually online right now, right? We are talking in that. So what it has to do with precision medicine? Soon you will realize it has everything to do with precision medicine. You go for a walk, you run, what you eat, you enter in your mobile, it takes the data and you are giving your database. On that database, someday they will be deciding your lifestyle and your medication, right? So it's all about data game nowadays. So with this, we will start our discussion. The first, we will discuss a concept of defining a disease. Then we will go for a modular representation of human disease, which is sometime a short note, and we have no idea what they are trying to ask. We still write something. That is a rule of theory exam. Please write something, right? Then we will touch upon system biology and precision medicine and application of precision medicine. So here you are actually dealing with definitely two short notes. One is the modular representation of human disease, and the another what you are dealing here is your system biology. The third one you are dealing is a precision medicine. So these are overlapping concepts, and you will realize soon that how it is going. So let's start our discussion now. Concept of defining a disease. If I ask you what is disease, and the question comes, okay, rheumatoid arthritis is a disease, hypertension is a disease, right? A condition. That's something, it's there in our concept that, okay, 1, 2, 3, this BP measurement I got, I will define my patient to have a disease called hypertension. But is it easy to explain? This is primary, secondary hypertension, secondary renal, cardiac, endocrine. There are so many things inside that, right? So initially it was just about hypertension. Then we are understanding the things. So what do you call a disease? Still, it is a difficult definition. In a true term, a disease is a condition, kind of you can label it as per the criteria and all. While illness is something what patient is suffering from, right? The illness helps us to label a disease. How confident are you at defining disease? Sometimes it is very difficult, right? That what exactly, you know, it is infection, but which infection? No idea. You all workup is negative. Patient is responding to antibiotic. It is some connective tissue disorder. Which connective tissue disorder? No idea. But see, before telling that no idea, you have to master the art. You have to master the information what is already existing. Then only we talk about something called no idea, right? Even after a good experience, do you see unexpected presentation, right? That happens sometimes that we read key, okay, diabetes presents like this, malaria presents like this, scrub typhus present like this. But then you see one patient who came with opsoclonus, myoclonus, and someone told, "Go, this can be seen in sudden onset in scrub typhus also." And then there are so many case report of that, right? So the odd things always happen. We have to be ready for that, because what we know till now is in the book by the people like us who have put everything in the book, right? It is the data what we are telling to the world, and someone is writing book out of that. It does not tell we are telling 100% thing. So that is something a difficult part. Do you even get irritated with changing nomenclature of lymphoma, neurodegenerative disease and all? What I read 5 6 years back, now even if I see Harrison, the things are completely changed. And the worst thing in our part happens, we ask MCQ out of that, right? So that is a thing. So this is a concept of defining disease. Now let me ask you a two very important question. What is your thought on this? You just keep it to you. Later on we will discuss. My first question is, where does rheumatoid arthritis starts? Okay, you can write your answer, you can write in comment, and later on we will discuss that how actually it happens. The second question is, atherosclerosis an immune disease? Yes or no? Right? We have learned atherosclerosis too much, but is it an immune disease or not? So have we given even thought about this, that what is actually happening? If I was asked where does rheumatoid arthritis start during MD, I would have easily answered that mostly it starts in metacarpophalangeal joint or the wrist joint, and later on, and sometime it can happen start in lower limb also. So my answer is joint, but it's completely wrong answer. It starts somewhere at mucosal level in respiratory or GI tract. Then the things goes bad, and this is finally manifest in the joint, right? So it is unpredictable things. More and more understanding we will solve. Atherosclerosis immune disease, yes. If you want to explore more, I have taken one class on this. You can ask for link, or it's already available in our app, that it is actually an immune phenomenon which leads to ultimate outcome of atherosclerosis, right? So these are evolving concept with our understanding. So that's the first defining a disease. Now let's talk little more. That in 18th century, it was simple. A patient is sick, he is having disease, over, no much discussion. 19th century, they told, okay, patient is having disease in this and this thing. We got the microscope, and then we told, okay, it's a granulomatous disease. Then in late 19th century, they got microbe also. Oh, this is Mycobacterium tuberculosis. So this is tubercular disease. Then we got, okay, at molecular level also, there's an interferon related disease and all in the late 20th century. And now we are having a challenge that what exactly it is. Is it just a microbiome causing disease, or a lot of interaction of host, environment, and all this microbiome which is causing this problem, right? So we are now getting confused where actually to take call, because now there is a lot of information, right? So initially ignorance was the bliss. Now the too much information is the problem, right? That's how the medicine keeps evolving, and that's how we have evolved. With, if you see, every 50 century the things changes. What we are dealing right now, 2050 is going to be a different picture, and we have to be ready for that. There is no alternate, right? We have to be ready if we are practicing. This is what we were talking, modular representation of human disease. Okay, so why we are talking all this part? Because before we come to this precision medicine, we have to build up a background what actually disease means. Then your precision medicine can crack it or solve it. Okay, so if we talk about this modular representation of the human disease, the first thing what you can see is a classical Mendelian disorder, a single phenotype, right? The patient has particular gene mutation or particular risk factor, and then he got the disease. And almost all patients with the same that disease will have same features. If you see Marfan syndrome, almost all will be same. Achondroplasia, almost all will be same with a particular mutation. Now you come to the another disease, which is a multiple phenotype. You see in sickle cell anemia, one has sickle cell disease, one has sickle cell trait. So that kind of combination or the presentation can come. Then you see the other disease where can be mutation in various genes, but the ultimate outcome is same. The phenotype is single. It's a hypertrophic cardiomyopathy. There may be so many mutations, but the ultimate result is a hypertrophic cardiomyopathy. Then you have a polygenic disorder like hypertension. There is no specific gene, but multiple gene, little contribution, and patient gets disease. Then you have a polygenic disorder with multiple phenotype. Ischemic heart disease. Some people will have stable angina, some people will have unstable angina, some people will have STEMI, some people have a non-STEMI, right? So that keeps varying. Even though the risk factor were same, the phenotypes are keep changing. And there are certain environmental disorder like corona, what we got, subacute bacterial endocarditis, we got dengue, we got. So this all are the disorder which comes in that category, that the disorder can change as per the phenotype also, right? So this is what we say a modular representation of the human disease. Now from tomorrow, any disease you think, try to fit in one category. Okay, what I am dealing with, because according to that we can plan the study of genomic, then transcriptomic, then metabolomic, proteomic, and the metagenomic. All these terms, omic are going to change the world. So we are going to have some idea about this omics. Okay, the omics will make the precision medicine. But one way we have to set our disease, that where actually it may fit. So this seems simple, right? Let me make it little more complicated, that what happens further. Okay, so few example before we go. What is convergent phenotype and what is divergent phenotype? Okay, this were not there in your previous editions. These are new things. So convergent phenotype is something where there can be the multiple or the different things, but the ultimate outcome is same. Like if patient has this sarcomeric protein mutation, hypertension, aortic stenosis, Fabry's disease, Pompe's disease, the ultimate outcome comes is a hypertrophic cardiomyopathy, right? Same way, patient can have various etiologies like TTP, HUS, malignant hypertension, scleroderma renal crisis, HELP syndrome, APLA. They all can lead to a common outcome of thrombotic microangiopathy. So they all are converging to the single thing, that is what we take convergent phenotype. Now I will make MCQ that all of the following are convergent phenotype except, right? So this is what we are scared of with new information. Then you have a divergent phenotype. One disease, but presentation can be quite different, and we need to understand that part. So the aortic stenosis either can have syncope, can have heart failure, can have angina pectoris. Antiphospholipid syndrome sometimes can present with stroke, sometimes with DVT, sometimes mesenteric thrombosis and the Budd-Chiari syndrome, sometimes MI, sometimes livedo reticularis, right? So everything this leads to this as a one diagnosis sometime, while multiple factors lead to this diagnosis. So this is called convergent and this is called divergent phenotype. This all are new information, better to know, right? So this is how, see, first we had idea of modular representation. This is few example that how modular representation, further we are refining to convergent and divergent phenotype. Now let me go a little further, how actually this happens, right? So this is what you have, the HBS, that is your sickle cell part, right? Then it can have effect of few more things, G6PD gene, HBF, HBC, beta thal, TGF beta, and all this mutation. And ultimately, there can be other environmental determinants like infective agent, hypoxia, dehydration, and all. And patient will have various pathologies like apoptosis, inflammation, immune response, thrombosis, resulting in one of this thing as a manifestation of sickle cell disease, right? So you see how things interact. The main problem was here, but this problem can be modified by multiple things, and this is how our understanding of disease is developing. For some patient the target will be this, for some patient target will be this, for some patient target will be this, for some patient it will be all target. How do we decide for which patient what target? We need more info, and that's what precision medicine is going to provide us, right? So once we have all the data, we will analyze with the supercomputers. We are like, we no hopes with the human brain there when we have so much of data, right? But we will just command it what to analyze. Let's hope we are still in power, right? So this is how the things are going to come, and this is how the things are going to manifest. So this is quite an important example to know. Now let me complicate it a little more. This is still okay, right? Brain like us can understand what we are talking. But now what about this? I have given up. I don't want to understand what is happening here, right? So many links, so many diseases and all. If you give this data to supercomputer, then only the things are going to run. So what we keep talking every day in the world, artificial intelligence, yeah, this is where we need, and actually it is in process already. We are talking a little late even, I can say, right? So this is something that they can actually with deep learning analyze this much data, and we can get multiple correlation or the outcome that what exactly might be happening in this processes and where my patient stands in this processes. Okay, so this is where we are building up. We started very simply that what is disease and how it is evolving over years. Then we went to the modular representation. Okay, this kind of disease, this kind of association and everything. Yeah, fine, that was a step 2. Then we went little complex, okay, to understand the example of this, and this is beyond our understanding, where we need computers, right? So this is something, as the information builds up, as the understanding progresses, we have to accept the fact we need help from each other, and definitely from computer, that what will happen next, right? So now this is what the exit, the disease component comes. I will come to this diagram again, but before that I just want to understand one more time what is system biology. Okay, so it is a method to study environmental effect on the health. Now see, let's imagine one disease, and I will bias it with rheumatology or something, that a person has rheumatoid arthritis and he asks you, doctor, why did I develop rheumatoid arthritis? And you start telling ki, don't ask me all this question, I don't know even. And that's a very honest answer, right? We don't know why he developed rheumatoid arthritis. But then we start developing some sense. Okay, there can be certain genetic factors which makes it susceptible, and there can be other environmental factors. So now you are telling me I was born with something that I was susceptible. I did not develop the disease at age of 40, and now at age of 41 I'm having this joint pain and all, right? So that's again a question. I was born with that, but it never developed. Okay, it's a slow process. So what was happening there, right? So we have a genetic problem here. So we can find, okay, we can do the genome study and we can find exactly what mutations are there. And if I, let me tell you some example which are asked in MCQ: PTPN22, PADI4, PADI4, STAT4 mutation. All this mutation you will read in rheumatoid arthritis. Some HLAs association you will read in rheumatoid arthritis. Yes, fine. This is the chance of RA increases, does not mean all the time it manifests. Now what does this gene do? The gene actually gets, DNA gets converted to what? RNA. RNA get converted to what? Protein. Yeah, that much we know. So now what happens is this genome is going to get transcribed, which is called transcriptomic, or the mRNA, messenger RNA, which are the ultimate message which makes protein. So when we do the genome DNA studies, it's called genomic. When we do the RNA study, messenger RNA study, it's called transcriptomic. But all the gene, how they will express, how much they will express, depends on epigenetics. Now, what is this epigenetics? It is the environmental influences on the gene to decide the expressibility, right? Let me tell you one simple example. All we have a common book called Harrison, right? So all we have is gene. But how do we read it? How someone guides you? How do you sincerely read and correlate will actually determine how much Harrison you know, right? So that is your transcriptomic, and which will make your practice. That is the protein. It's a proteomic, right? That's a simple example. So this environmental cues play very important role to decide how these genomes are going to express in RNA, and this RNA is going to make proteins. Now, study of proteins are called proteomics. Now, what is the function of proteins? Metabolism most of the time, and structure, synthesis, and all things. So what it makes or what it does function, if you study that, it's called metabolomics. Now, one more component we already have discussed is the microbe. So microbe study is called metagenomic, right? So the microbe also depends on what I eat, where I live, how much stress do I take, how much do I sleep, what are my company, what are my friends, everything decides to the certain extent a microbe, right? That is very important. How many antimicrobial I have taken, breastfeeding for how much time it was, everything depends, sir, whether I'm a preterm or whether I'm a normal term, everything determines this microbe. So the answer is not straightforward. certain genomic factor with the epigenetic influences which result in transcriptomic, ultimately the proteins and the metabolism, which again can get influenced by the microbiome. Everything decides whether we have susceptibility of disease or not, and this is called system biology, right? So this is a concept of system biology. Now if you know a disease, you have to ask a question. Do you know genomic of hypertension? Do you know epigenetic of hypertension? Do you know transcriptomic of hypertension? Do you know proteomic of hypertension? Do you know metabolomic of hypertension? Do you know the dysbiosis of hypertension? And with this, you will make a model that what exactly is the problem is, right? So actually you can make a biomarker accordingly, and you can tell, okay, this patient lived in this environment, has this many changes in his blood or the other organ, is likely to have this disease and likely to respond to this antibiotic or this medication for his disease, right? This is called system biology going to precision medicine, right? So this is again, this is how we live. It's a human genome. Then there is early immune and metabolic programming at the embryo and the fetal stage. Then you have certain things like food diversity, food, tobacco toxin, screen time, UV light, stress, psychosocial asset, and then there are other things affecting, and then there is a geology, right? So everything is a system biology. So if you think the disease is purely genetic or certain genetic risk factor, you are not thinking enough. You have to think very wide. Okay. For theory exam, for treatment, what we have, we have to treat it. Okay. So that's how. But see, it's something, be open to concept. Don't accept so fast, right? Now, this is again what you tell that how the things happen. You have a genomic work, then transcriptomic, then proteomic, then metabolomic, then psychological network, which ultimately decide the clinical phenotype. And that also depends on environmental exposure and microbial interaction. With improved understanding of disease, this all result. Complex disease reclassification we can do, disease prevention we can do, and network targeted therapy we can go, and we can go to precision medicine. Now let me tell you that irritation part. The lymphomas nomenclatures are revising because of all this thing. We are understanding it. Even we have disease called systemic sclerosis. Now we have kind of a new classification of that. I am not even going into that part, that what is happening exactly, but yes, things are changing. Okay. To the extent it is changing that if you actually analyze like that, you can actually decide very early what drug is going to work for my patient. And believe me, if there's all validation of the studies, and if we have enough money, I will definitely try to go for that, because that works. It's a proven thing. My hypertension is different from your hypertension. I may respond to CCB well, you may respond to ARB well. How do we know? That is precision medicine, right? So hopefully someday we know. Fine. And hopefully someday only we know, right? Otherwise we will be out of business. Fine. So now this is a new term. It's called reticulotype. Okay, we have heard of reticulocytes. Now this is reticulotype. So now reticulotype actually integrates your genotype and phenotype depending on information from the multi-omic. Now you understand multi-omic, right? It's a DNA, genomic, RNA, transcriptomic, protein, proteomic, metabolite, metabolomic, microbiome, metagenomic, and clinical exposure. That's an environmental effect. That's epigenetics, right? So this all multi-omic will tell you that this is kind of your network. And depending on your that, you get the reticulocyte and you get your individual therapy accordingly. Individual 2 might have a different things, might get a different drug. 3 might have a different thing, might have different drug. So everyone doesn't deserve dolo, right? Fine. So it's not everyone gets steroid, everyone gets derifylin, and everyone gets Dexona, right? It is not like that. It's not everyone gets ceftriaxone. Fine. So it's a different drug for different people. But that is a dream. Hopefully I can call it a good dream. Fine. But this is how the things are going on. This is called reticulotype. And I was also not aware of this term before Harrison 21. This is actually given there. Okay. So this is how precision medicine comes in picture. It can help us in drug discovery also. See, we have understood the disease, that this is genetic, on that this transcriptomic and then protein changes happen. At what point, if I do the target, the disease can change more. So this is how we can even do the drug discovery. And many of the drugs are result of all this part, right? You have TNF inhibitor, you have IL-17 inhibitor, you have a specific target in cancer therapies. it comes out of this. And it's actually beautiful thing because this all are computational analysis of the models, right? So it's not you have to do all the time this murine experiment and come with a failed drug. Here from starting we will get the data that this is the most likely drug. So instead of starting from 1,000 molecule, we will start with 3 4 5 molecule from the preclinical study of a mouse model. Then if it's doing well, then we will go to 1 2 3 4. So you just imagine the speed of discovery of drug. And that's why we are getting so many new therapies in cancer. And we rheumatologists are very smart. We let them discover, and with the lower dose, we start using it in our diseases because we have some immune problems there, right? So that's how the things are going. It's called repurposing of the drug or using it, the concept. Methotrexate, it was a cancer therapy. Rituximab, it was a cancer therapy. Nowadays, we use it every day. So we have adapted almost everything from cancer. Okay, so we are good at those part. Fine. So this is the drug discovery, which also can happen with the precision medicine. And this is what we say the future, that you have your omic data at a particular time point. They will take your exposome or the social determinant data, right? We are always exposed. Right now we are on mobile screen. I don't know who is listening to us even, right? So everyone knows where we are moving around, and suddenly at the month end, Google will send you, this was your year look like. And we were, oh, you know that, right? So this all can go like that. Then you have a like study participant generated data, that we have all this watch and all, where our all health activities are getting monitored, and there are so many apps we are active on, and the microbiome. So this all will determine that, okay, what exactly dynamic phenotypes are there and what time you deserve what drug, and this is what is going to tell with that, right? So with this I will conclude this class. And this is a really important slide. Will human touch become obsolete? I think we will survive, but next generation, I am not sure what will be their medical practice, right? So ultimately, summarizing the part of your precision medicine, you have to be very simple on that. That first mentioned concept of disease, how it evolved, what is the modular representation, and few example of convergent and divergent. And then it is too complex to understand, so we need all the level of system biology. Try to explain the concept of system biology and how actually it is going to help us here with improving our practice and all further, right? And reticulotype concept you can write, and drug discovery help. And this is how we will going to complete. Are we ready for future? We don't have option, right? We have to be ready for future. And this is the last slide, and this is very interesting data. Okay, that this 10% in 1950 somewhere was the screen time. Then in 90s it went to 30%, and now I think we all agree, for me I will say 80% of my time goes to screen. Okay, it's a bad thing, but that's what happens. When I'm seeing patient, I'm entering data on my laptop or computer. Otherwise, I'm reading on laptop or computer. So it's always like that. We are stick to screen, hardly 20-30%. And someone will tell, okay, when I'm having food, also my mobile is like I'm doing like this, right? So this is something, you earn your money on internet, you spend your money on internet, and you depend on your digital avatar. We can't deny the fact, right? So with this, I will conclude this class. Thank you. If any doubt, hopefully no, you can ask and you can directly message me. Thank you.